Kingdom, Animalia. The original ancestral animal. The animal kingdom probably evolved during the Cambrian period, 540 million years ago. The earliest animal fossils are from the late Precambrian period, 600 million years ago. This is a preserved fossil of Dickinsonia. It is believed to be one of our oldest ancestral animals, and they actually discovered cholesterol molecules in the fossils of these Dickinsonias. Cholesterol is a hallmark of the animal kingdom. The Cambrian Explosion. At the beginning of the Paleozoic era, multicellular animals underwent an explosion in diversity known as the Cambrian Explosion. This is an artist's rendering, based on fossil evidence, of some of the bizarre life forms now extinct that emerged during this time. So in the whole fossil record up until this point, all we saw were just a few basic animal body plans. And then all of a sudden, within a very short period of time, geologically speaking, all these new animal body plans evolve. All these new animal phyla, including all the ones that we see today, and the ones included here in this picture that are now extinct. The Cambrian Explosion. All animal body plans that exist today can be traced back with geologic and fossil data to the ancestors of this time period. Over time, natural selection favored some traits over others, and today we are left with a small percentage of the body plans that existed in the Cambrian period. Diversity of the animal kingdom. It is easy for us to appreciate biodiversity by studying the animal kingdom. There are more than 1 million species of animals described so far, and it is predicted that tens of millions more have yet to be discovered, many of them insects. Animals can be microscopic like Hydra, small like a ladybug, or large like a blue whale. They can have hard shells like a snail, or be so soft that they appear translucent like jellyfishes. They can spend their lives attached to a single surface like tube worms or fungi, drift in water currents, or be mobile like a giraffe. Animal characteristics. With the great diversity in this kingdom, it is important to point out that there are characteristics shared by all its members. All animals are multicellular heterotrophs. Animals must eat other organisms to obtain their energy. Animal cells are supported by their cytoskeletons and by collagen. Animals do not have cell walls like plants or fungi to give rigidity to their cells. Collagen forms strong and flexible rope-like structures that help support cells. Animals are diploid and usually reproduce sexually. Most animals have Hox genes. Homeotic Hox genes define the head-to-tail pattern of development in animal embryos. Phyla of the animal kingdom. The animal kingdom can be broken down into 36 phyla based on body symmetry, tissue layers, and developmental patterns. Some of these phyla include sponges, jellyfish, flatworms, roundworms, mollusks, segmented worms, arthropods, echinoderms, and chordates. Symmetry. Animal bodies can either have radial symmetry or bilateral symmetry. Radial symmetry is a common feature of simple animals. Radially symmetrical animals have all body parts radiating out from the center of their body, such as this Hydra. Bilateral symmetry is more commonly found in complex animals. Bilateral animals show a right and left side, such as this butterfly. Symmetry. For echinoderms, radial symmetry is adaptive. There is evidence suggesting that echinoderm ancestors are bilateral. Tissue layers. Animals with bilateral symmetry are triploblastic. They have 3 tissue layers. The ectoderm is the outer layer. It develops into the skin, brain, and nervous system. The mesoderm is the middle layer. It develops into the internal tissues and organs. The endoderm is the inner layer. It develops into the lining of the gut. Radial animals are diploblastic. They lack the mesoderm layer, so they lack complex internal organs. Tissue layers. This chart summarizes the tissues that are present across the animal phyla. Notice that the sponges do not have any tissues. Then the cnidarians, which include jellyfish, are diploblasts, so they just have 2 germ layers. The organisms that appear after the jellyfish on this cladogram have 3 germ layers. So that includes the flatworms, roundworms, mollusks, annelids, arthropods, echinoderms, and chordates. All of those phyla have 3 germ layers. Body cavities. Animals with 3 tissue layers, the triploblast, can be further subdivided based on whether their mesoderm develops an internal body cavity. A body cavity is a fluid-filled space within an organism. In animals, the main body cavity is called the coelom. It is located inside the body's outer covering, and it surrounds and contains the digestive tract and other organs. Triploblasts can be classified into 3 categories based on the presence or type of body cavity. Acelamates, such as the flatworm, have no body cavity. Pseudocellular, such as the roundworm, have a partially lined body cavity. And the celamates, such as the earthworm, have a fully lined body cavity. Acelamates. Acelamates have only a digestive cavity with no lining. So in the example of this planarian, we have the digestive tract here. That's where all the food goes through. And then between the outer body wall and the digestive tract, it's all just solid tissue. There's no space in between, so therefore it is an acoelomate. A, meaning lacks a coelom, lacks a body cavity. Pseudocellular. Pseudocellular have a partially lined body cavity which loosely holds the animal's organs in place. So as we can see in the diagram, there is a muscle layer just on the inner part of the body wall, but not actually surrounding the digestive cavity. So we have a partially lined body cavity that loosely holds organs in place. Celamates. Celamates have a fully lined body cavity called a coelom, which holds the animal's organs in place. As we can see in the diagram, there is a layer of muscle that lines the inner body wall, and it also lines the digestive cavity, and it connects by the mesentery. So the advantage of being a coelomate is that you could have more organ systems evolve and be held in place as the animal moves. Body cavities. In this cladogram, the triploblasts include the acoelamates, pseudocellular, and celamates. Acoelamates include flatworms, which have no body cavity. The pseudocellular in this cladogram include the roundworms, which only have a partially lined body cavity. Then the true celamates, which have a body cavity that's lined with mesoderm, include the mollusks, annelids, arthropods, echinoderms, and the chordates. Developmental patterns. Triploblasts can be classified as protostomes or deuterostomes based on key differences during embryonic development. The first opening of the digestive tract. In protostomes, the mouth is formed first and the anus second. This order is reversed for deuterostomes. Gut cavity formation. In protostomes, the mesoderm splits to give rise to the gut. In deuterostomes, the gut is formed from pouches in the mesoderm. Cleavage pattern. Protostomes undergo spiral cleavage of the early embryo. In deuterostomes, the embryo experiences radial cleavage. Developmental patterns. This diagram illustrates the differences between protostome and deuterostome embryonic development. In spiral cleavage, the cells kind of divide in such a way where they're not directly on top of one another. They're kind of like at angles with each other. That happens in protostomes, and in deuterostomes we have radial cleavage, and in this case the cells are sort of more stacked on top of one another. Then during gastrulation, when we have multiple layers of cells, the blastopore, which is the opening, becomes the mouth in the protostomes, but then that blastopore becomes the anus in the deuterostomes. Developmental patterns. In this cladogram, we see that protostome development occurs in the flatworms, roundworms, mollusks, annelids, and arthropods, and deuterostome development occurs in the echinoderms and chordates. Note that echinoderms, which include sea stars, sand dollars, and sea urchins, even though they are radially symmetrical, they are more closely related to the chordates than they are to, say, the cnidarians, because they have embryonic patterns of deuterostome development.